Hello all, welcome back to Nanotech. Today's topic is Nucleic Acid Nanosensors. Here I will explain in details about basics of DNA, types of nucleic acid binding interactions in biological systems and nanosensors designed for detecting DNA binding interactions. Basically, deoxyribonucleic acid, DNA, which is a polymeric chain made up of units that repeat themselves and are known as nucleotides, conveys significant genetic instructions that are essential for controlling biological processes in living cells. Mutations in DNA have always been correlated with various diseases including cancer. DNA typically exists as two strands of oligonucleotides wrapped around each other to form a double helix. The double helix structure of DNA is characterized by two grooves which allow binding with a large variety of proteins or drug molecules. Proteins that bind to DNA, such as nucleases, enzymes, and transcription factors, control the biological activities of DNA, typically gene expression. The primary target of many pharmacological compounds that interact with DNA in different ways in order to alter its function. The advancement of nanotechnology has contributed significantly in developing novel nucleic acid-based biosensors involving the use of different types of nanostructures. These nanosensors commonly use plasmonic nanomaterials such as gold and silver nanoparticles, fluorescent nanomaterials like quantum dots and carbon dots, and electrochemically active materials like carbon nanotubes and graphene. The interaction of DNA with nanomaterials are very important for biosensing applications. The uniqueness of a nanosensor lies in the similar length scale of the nanostructures to the biological targets to facilitate better binding interactions mimicking natural interactions, which may further allow in vivo applications. Additionally, nanostructures' high surface area to volume ratios allow a variety of biorecognition components to bind and collect the target analyte, greatly increasing the detection sensitivity. Types of nucleic acid binding interactions in biological systems. The nucleotides typically composed of a 5-carbon sugar called deoxyribose and a nitrogen-containing base, which are connected through phosphodiester linkages within a strand. The bases include thymine, cytosine, guanine, and adenine. The secondary structure of DNA is a double helix with two antiparallel polynucleotide chains firmly held together by hydrogen bonds, where A forms 2 hydrogen bonds with T while C forms 3 hydrogen bonds with G in the double helix. The sequences of the strands are complementary to each other. The interactions of biomolecules or drugs with DNA double helix are varied. Typically, proteins interact with DNA through specific base pair recognition, multi-specific binding, or non-specific binding independent of DNA sequences, whereas drug molecules can interact in covalent or non-covalent interactions. Protein-DNA binding. Proteins that possess DNA binding domains can bind to DNA through non-specific, specific, or multi-specific interactions. Drug DNA binding. DNA, especially in its double-stranded form, can complex with many natural or synthetic molecules, therefore DNA has become the pharmacological target of many drugs. Aptamer target binding. Aptamers are single-stranded DNA or RNA that can bind to their targets with high affinity and specificity like the antigen-antibody interactions. Aptamers, often 25 to 90 bases in length, are derived from an in vitro selection process called systematic evolution of ligands by exponential enrichment. Nanosensors designed for detecting DNA binding interactions. To detect DNA binding interactions, such as protein DNA binding, drug DNA binding, aptamer target binding. Among which, the most promising one is the nanosensor design, which uses either the plasmonic nanostructures for direct optical sensing or employ various type of nanomaterials such as graphene, metal oxides, rare earth metal, magnetic nanoparticles, etc. to achieve signal amplification or enhancement in sensing performance. Particularly, optical and electrochemical detection principles have been widely applied in developing nanosensors for various analytes. Metal nanoparticles-based colorimetric detection. Colorimetric sensors are simple, low cost due to their easy readout, which is even possible by naked eyes detection. Noble metal nanoparticles such as gold, silver are suitable colorimetric indicators due to their excellent extinction coefficients and unique distance-dependent optical properties. In colorimetric detection, aggregation strategy has been employed successfully to detect not only the sequence-specific protein-DNA interactions without any tedious surface modification steps, but also provide useful insights into their intrinsic binding mechanisms in homogeneous solution like that in the biological systems. Surface-enhanced Raman spectroscopy (SERS) detection. SERS is one of the most promising technique for molecular detection which merges the benefits of molecular fingerprint specificity with single-molecule sensitivity. SERS have been employed to study protein DNA or drug DNA interactions, achieving enhanced signal and therefore improved detection sensitivity. Thank you for watching. See you soon with another interesting topics.